Welcome to the International Conferences on Toxic Cyanobacteria, ICTC. This particular conference is special for me because we're back in Toledo, Ohio, where the very first one was in Dayton, Ohio in 1980. So my first slide is just nostalgic back through the ages 1980 to 2022 to bring you up to date. For those of you that weren't born in 1980, this will be particularly good. We had the very first International Conference on Toxic Cyanobacteria supported by the US EPA in 1980 here in Dayton, Ohio at Wright State University. There was a hiatus before Jeff Codd managed to organize what we've referred to as the second ICTC in 1993, and we had that one in, of course, in Bath, UK. From then on, a fairly regular program, so 1995, 1998, 2001, 2004, 2007, 2010, 2013, 2016, and then the last one, 2019, the 11th, in Krakow, Poland. So here's a slide from the very first one, June 29, July 2nd, 1980, Wright State University. There aren't very many people here that even I recognize, although there are a couple. There's Maria Runegar, who's very well known for some of the initial work in Australia. Valerie May on the left side in the blue dress is quite an old timer. She did some of the very first work in the 1950s on toxic farm pond animal poisonings in Australia. And then third from the left, was Paul Gorham. You can just see his head. He was the initiator of much of the current work on toxic cyanobacteria when he was in NRC in Ottawa, head of the plant physiology division. And that's where we managed to get the initial work done on toxic cyanobacteria in Canada, and also out of his work came ASM1, artificial seawater McLaughlin, which became the standard media for being able to culture cyanobacteria in the lab. So with that introduction, the second slide will bring us back up to 2019. And here's a much better slide. Darius is on the right side next to me there. He organized, along with his organizing committee that you see the names of there, May 5th to 10th, 2019. And that was an extremely good, represented conference. So those are the conferences that bring us up to the current time period. So for my talk, I'd like to sort of take a broader view and a sort of more expansive futuristic view and talk a bit about how we might move from monitoring and management of cyanobacteria, harmful algal blooms, to prevention and remediation, which I think is timely and important, and I'm sure that many of you would agree that that's a good topic. So with that introduction, I move to the next slide, and this slide is to sort of emphasize how unique, important, and interesting cyanobacteria are. We all talk about how cyanobacteria are some of the oldest life forms in the fossil record. We refer to them as oxygen-producing organisms important for development of the Earth's atmospheres. Well, in Science, April 15th, volume 376, some new information came out which further sort of places the time schedule for cyanobacteria. This is in the Barberton Mountains in South Africa, evidence for some of the Earth's first land ecosystem in a 3.2 billion year old rock formation. And out of that study, the statement in Science says that the genomic record is independent and consistent with the idea that those were early ancestors of cyanobacteria. So whether you talk about 3 billion or 3.5 or 4 billion, we now have a sort of a timestamp of 3.2 billion that you can use in referencing how long we've had cyanobacteria. So as a consequence of that, you can imagine cyanobacteria have had a long time to sort of work their ways in our ecology and ecosystems how that is. And there's no question that, in the early, fossil record, there were blooms, and we've had blooms for a long time. The current issues with blooms have more to do with how we manage our water supplies. And this slide shows what is an obvious sort of trend, and that is that CyanoHABs or cyanobacteria harmful algal blooms are increasing. So this comes out of some work that the database is from the US EPA, and Bluefield Research has taken that database and created some interesting statistics. And the title of this is that through increased monitoring and higher levels of phosphorus, along with nitrate and increased temperatures, harmful algal bloom events in the US have grown over 7-fold in the last 10 years. HABs have substantial direct and indirect economic and health effects on surrounding populations and are drivers of increased attention and regulation around nutrients involved in agriculture like phosphorus and nitrogen. So 3 sort of data points that people like to point out. For example, the Florida Department of Health has estimated yearly HAB-associated costs of around $60 million. The 2015 West Coast bloom of Pseudonychia, which is not a harmful algae, but again, is not a cyanobacteria harmful algae, but just a marine harmful algae, a diatom, estimated to cost the Dungeness crab industry $48 million. And then the recent HAB occurrences in the southwest corner of Lake Erie are estimated to exact an annual economic cost of around $70 million related to tourism and fishing. So there you see the graph, roughly a linear increase in harmful algae blooms from 2010 to 2019. What my title refers to, in the next 50 years, an opportunity for remediation, prevention of cyanobacteria harmful algal blooms, comes after roughly 50 years of my own research in this area. And what I'd like to do is to point attention to a refocus of away from monitoring and management to actual remediation. So to accomplish that, let me start by a little background, which says, as the next slide shows, CyanoHABs are problematic. I think we all agree with that, because it's been just over 50 years since the term eutrophication became part of the common language in referencing cyanobacteria blooms and water quality in the Great Lakes, and of course, Lake Erie in particular. And that's what the slide to the left shows, an early CyanoHAB bloom in Lake Erie, which we now think was the non-toxic Phanozomenon, Flos aqua. And then ever since that time, much effort and expense has been spent on HAB monitoring detection, which has given us a very good perspective on the occurrence and distribution of both organisms and their toxins, which we now refer to as cyanotoxins, recognizing that there are a lot of other secondary metabolites, but it's really the cyanotoxins that have been the focus for research. But we then want to make the definitive statement that this monitoring and responding with after-the-fact response methods to CyanoHABs has not led to the development of sustainable solutions, even after 50 years. Instead, what we have seen is largely a growth of an industry involved with consultants and treatment companies providing reactive symptomatic treatments for the symptoms of eutrophication and CyanoHABs. So what we're left with is somewhat of a paradox that we might call symptomatic treatments, where our toolbox of methods act to actually accelerate eutrophication, making the situation worse in subsequent years, which in reality tends to drive more business only for the continued symptomatic management of these blooms worldwide. So my argument is that the next 50 years should be about prevention and remediation. And I would like to propose that we can accomplish that through an understanding, or at least a better understanding, or a better focus of why CyanoHABs dominate, combined with what I call a multi-level mitigation approach, using the existing management methods, but combining them in a more scientific fashion that involves air-water flow, complemented with bioaugmentation that reduces and removes lake sediments. The consequence of this treatment process tends to increase water volume, move nutrients into food webs that are beneficial, and allows competition by beneficial algae to actually outcompete harmful cyanobacteria and reduce sediments that would otherwise encourage the growth of aquatic plants. My next statement would be to clarify what I mean by monitoring, management, and mitigation. We call it perhaps the 3 M's. We now know that monitoring really involves remote observation, sampling, taxonomy, cyanotoxin detection. All of these tend to inform the process of setting HAB advisory guidance, management, and mitigation. And again, we've done a pretty good job of that. Management and mitigation is really an attempt not to prevent or remediate, but only to reduce or temporarily control existing CyanoHAB effects. And these approaches do serve to minimize bloom effects, but if we really want true prevention, it involves maintaining high water quality, and then failing that, which we have done to date, prevention involves remediation. Let's just do a quick review of the main ways that we do management and mitigation. There are 3. Number 1, physical, where we manipulate lake intake location or water intake location and depth using aerators to kind of de-stratify, and mechanical mixers for long distance circulation, and even barriers involving sand and carbon. The second way are biological controls, which we actually manipulate lake ecology to favor cyanobacteria grazers, kind of a top-down approach, and increased competition for nutrients, sort of a bottom-up approach. The third way that we do management and mitigation is with chemical controls, and largely this is focused on phosphorus. So we have the various phosphorus treatments like lime, aluminum sulfate, ferric chloride, even lanthanum and clay particles. This tends to, they are used to both bind nutrients and flocculate cells. We also use algicides like copper sulfate, hydrogen peroxide, potassium permanganate, and herbicides. My argument is that while these methods for management mitigation do give us the temporary, what we really need to do is to look more closely at how we might combine these methods in a more thoughtful scientific manner. And that's what I want to do. My reference for making these 3 physical, chemical, biological methods is at the bottom of the slide, showing a very recent scientific summary for policymakers in 2019 from IOC UNESCO. Well, why do cyanobacteria blooms become dominant? Well, I think really, while we might want to throw in various other things like flushing rates and global warming, it really comes down to CyanoHABs are driven by eutrophication. And that's a word derived from the Greek meaning of well-nourished. And of course, we tend to think of that as meaning an excess of nutrients, but the real problem stems from the competitive advantage that cyanobacteria exploit in eutrophic water bodies to become the most well-nourished organism in the ecosystem. We can expand that a little bit by looking at some of cyanobacteria's competitive advantages. We now know, even most recently from some fantastic work done in South Africa, showing that cyanobacteria in the fossil record go back at least 3.2 billion years. So we say that the cyanobacteria were one of the first life forms capable of light-driven photosynthesis. In these extreme environments, they needed to be highly adaptable, agile, and hardy to survive and proliferate, which they've done a very good job of. Now, over the millennia, they have retained many of these capabilities, which they now use to exploit nutrient availability. This enables cyanobacteria to outcompete and dominate. The competitive advantages they exhibit include nitrogen assimilation. They have specialized cells for nitrogen fixation. And By doing that, they can utilize all three nitrogen sources in the environment to help them exceed and do well. The second advantage they have is they have buoyancy control. They have gas vacuoles literally within the cells that they use to control their buoyancy, inflating and deflating gas vacuoles. This allows them to dive to the bottom or rise to the top, taking advantage of light, temperature, and nutrients. Third, they're able to mobilize sediment nutrient stockpiles. They can mobilize nutrients in the sediment, making those nutrients bioavailable in the water column, where they are used to produce more biomass. And finally, which tends to be the focus for much of the research these days, is their toxin production. Various cyanobacteria produce toxins that make them unpalatable and also used as defense mechanisms. These cyanotoxins may also have potentially fatal effects on aquatic life, terrestrial animals, and people. So as a final statement, we say in eutrophic conditions characterized by stratification and deoxygenation, as well as sediment nutrient accumulation, we can understand how cyanobacteria utilize simple tactics to leverage their competitive advantages and impose distortions on the nutrient cycle. Now, one way we can help explain why cyanobacteria are so dominant is to look at the basics of systems theory. Now, systems theory uses the term attractors to describe the main determinants of system behavior. So in a healthy lake, the main attractors are an oxygen-rich environment, a productive biodiverse food web, and achieving nutrient clearance from the system. Now, for cyanobacteria to achieve dominance in a eutrophic lake, these dominant attractors must be changed to an anoxic benthic environment, a constrained food web lacking biodiversity, and a nutrient recycling that supports algal and cyanobacterial dominance. One of the key researchers have used this systems theory to kind of explain how cyanobacteria dominate is Cottingham in a paper in 2015, where she and her colleagues used systems theory paradigm to describe and model cyanobacteria's ability to single-handedly transform a low nutrient lake into a eutrophic state within which they can bloom and dominate. So, with that background statement, I'd like to point out how we might move beyond management and mitigation. Again, mitigation implies removing CyanoHABs from a water body and preventing their occurrence, and it relies on a management tool approach. However, unless we are willing to make major efforts towards protecting and improving watersheds, in other words, watershed management, and address global warming, the best we can do is bloom management. Whereas remediation, which is what I'm proposing, utilizes this management mitigation techniques in a more multi-level scientific approach, an approach that returns the water body to pre-cyanobacteria HAB states. This involves, 1, understanding basic morphometry or bathymetry using things like sonar scans, then recognizing biology and chemistry of the water body in question. This informs the placement of a well thought out air-water flow system as opposed to just laying airlines in a lake and bubbling air. Secondly, we use that air-water flow and we complement that with bioaugmentation that together removes lake sediments, shifts the phytoplankton population to diatoms and green algae, which are more, of course, to our liking. These treatments increase water volume, move nutrients into food webs that are beneficial, allow competition by beneficial algae to outcompete those harmful cyanobacteria, and also tends to expose hard sediments that don't encourage the growth of aquatic plants, because often it's aquatic plants that we want to manage as well. So, a little bit better definition of bioaugmentation would be to emphasize that we're using native enzymes, not bacteria, to increase sediment decomposition, plus selective minerals and micronutrients that favor diatoms and green algae, allowing us to shift that phytoplankton population. In the case of the field examples that follow, the two products are referred to as an enzyme mix and a nutrient mix. So with that background, I want to quickly work through 3 examples, and that is to remediate cyanobacteria. The first example is an example of simple sediment removal. We actually are removing the sediment, therefore removing the nutrients out of the system, and then supplementing that with our bioaugmentation. This example is a small lake called Lake Peekskill in New York, has 60 acres, maximum depth at 26 feet. The focus here has been that this lake was closed to the public, and as late as 2017, it was only open for 5 days. So a remediation method was commissioned in June of 2018 when the lake was currently closed. And as of October 2019, showed a sediment reduction of over 49,000 cubic yards and had a sediment phosphorus reduction of 11%. And following the treatment, there were no confirmed CyanoHAB incidences in 2019 or 20, and in fact, that lake is quite productive and back to its normal fishing capabilities. This was accomplished first with a bathymetric survey, and in this next slide shows the lake actually, and we focused on the 14 feet or deeper depth line, and you can see in October 2019 scan, which is yellow, shows an increase of about 4 acres of that 14 foot or deeper in comparison to the similar scan in blue of April 2018. So what we have done is remove the sediments using that methodologies. We haven't had to worry about nutrient inputs. We've literally reduced the sediment, which in fact reduces the nutrients. Another way of looking at that is a side. Now you can see the blue line, which was the pre-treatment scan depth, and you can nicely see the red line, the 2019 scan profile, showing that depth increase, which gave us the sediment reduction and the depth increase. So that's quite a good example. It's in this case focusing on sediment reduction to show that in fact what happens after that is you get cyanobacterial reduction and consequently no harmful algal blooms after that. The second example is actually showing how this methodology reduces the cyanobacteria numbers or the cyanobacteria populations. And this is in Ohio, the city of Bowling Green and their water treatment, drinking water treatment. They draw water from the Maumee River, which in the slide you can see is just to the top left here. The point here is that this reservoir is relatively small, giving them only about a 28 to 30 day supply. So they constantly have to draw water from the river, which prevents them from drawing water only when the cyanobacterial blooms are minimal or non-existent. So they're really required, or they're forced to draw water at all times, including times when there are high cyanobacteria populations. So this study was done in 2016 versus 2017, and the numbers you're going to be shown here, week 31, which is midsummer when the bloom populations are highest. So in that top left, the 3rd of August 2016, you see that the phycological count in the river at that time was about 150,000 cells per mil. And in the reservoir, almost an equivalent amount of 124,000 cells per mil. The cyanobacterial populations at that time were about 68% in the river, 102,000, and almost exactly the same in the reservoir. In other words, no reduction of cyanobacteria. Post-treatment, July 2017, total phycological count in the river was about 160,000, but note the total phycological count in the reservoir using the methodologies were 41,000, and a reduction in the cyanobacteria population composed of about 42% down to less than 1%. So there's a good example showing actual reduction of cyanobacterial populations in the reservoir, even though you're drawing high cell counts of cyanobacteria directly from the river. The third example is to show scale. In other words, these other two treatments were fairly small water bodies, but now this one is a larger water body, actually 850 acres, depth 170 feet, Talvaca Reservoir in Puerto Rico. In this case, what has happened, if you look at the slide that just popped up, you notice that we're going to use examples of actual treatment chemical reduction. And in the process of doing this treatment chemical reduction and the methodologies, you also reduce the cyanobacteria and geosmins and other taste and odor compounds. Notice the coagulant saving. The lime savings of around 288 down to 120, and the chlorine savings from 140 to 76. Now, in particular, those chlorine savings are vastly important, not only for cost effectiveness, but also your trihalomethanes, total trihalomethanes, or your disinfection byproducts were reduced. And that's shown in this next slide. The TTHMs, the total trihalomethanes, are disinfection byproducts. The one on the left is showing a reservoir that was not treated, having 111, which exceeds the cutoff point of 80. And notice in the two within Talvaca, you reduced it down to 59 and 56, well below that cutoff level of 80, which is the guideline values. So you have three examples now, one showing actual sediment reduction using this methodology, a cyanobacterial cell count reduction, and a vast improvement in the chemical treatment as well as cyanobacteria populations. So in summary, the methodology that I'm proposing to pay more attention to and seems to work quite well actually uses a systems theory to help us really understand and guide our remediation, and allows us to understand cyanobacteria's competitive advantage and move on. Number two, studying the individual water body, followed by destratification, reoxygenation, and bioaugmentation. The bioaugmentation assists the sediment digestion and the movement of nutrients into beneficial components of the food web, but not CyanoHABs. We get an increase in depth, which is equivalent to a water volume increase or a sediment volume decrease, and we get a reduction in phosphorus concentration in the remaining sediments. Those biodegradable organic sediments are mostly generated within the lake and have increased over the last 100 years due to eutrophication. These sediments are susceptible to removal by oxygenation, and this is, in fact, the principal method in thousands of activated sludge waste treatment plants, which oxidize the mass of human organic waste to CO2 using fine air and vigorous mixing. And finally, in addition to the three examples I've just shown you, these multi-level methods have been successful in other Midwest lakes, Michigan, Pennsylvania, New York, Virginia, and as an argument can be applied worldwide. I should acknowledge that these lake result data come from a company called SIS Bio, and I think that it's fair to say that, at least from my perspective, this is a company that has done a very good job of using the systems theory approach and using those existing management methods, but applying them in a more multi-level scientific manner. Some of the key references that have sort of led me to these conclusions and presentation are listed right here. There's an awful lot of that sort of materials out there. There's not one of these that I would point out except the number 4 is the Cottingham presentation, Cyanobacteria's biological drivers of lake nitrogen and phosphorus cycling, and then some of the user manuals like water utility manager guides, etc. So I thank you. Thank you for your attention, and there's my email at the bottom if you want to send any questions or comments. I'd be happy to respond.